Microbial nutrition and growth. In this lecture, you'll learn about some of the environmental factors that influence growth of microbes, such as the type of nutrients, temperature, pH, oxygen requirements, and osmotic pressure. We will look at the cell cycle, how individual cells grow, and also growth of a population of cells. And then we'll take a look at some different methods used to measure population growth. Recall from previous lectures that cells are about 70% water. The majority of a cell is composed of proteins. Proteins are the most prevalent organic compound above carbohydrates, nucleic acids, and lipids. Also recall that 96% of a cell is mainly composed of 6 elements: carbon, hydrogen, oxygen, phosphorus, sulfur, and nitrogen. So cells must be provided with nutrients that contain these and other elements that occur in the cell in smaller amounts. Essential nutrients are those that must be provided to an organism for its growth. They must transport these molecules into the cell and metabolize them. Macronutrients, like proteins and carbohydrates, are needed in large quantities. They are required for maintenance of the cell structure and to carry out metabolic reactions. Although micronutrients like manganese, zinc, nickel, and iron are required in trace amounts or small quantities, they are extremely important for the function of certain enzymes and to help maintain the structure of some proteins. Pictured here is the hemoglobin protein, which requires the micronutrient iron to function properly. Organic nutrients are those that contain carbon and hydrogen. These molecules contain bonds between the carbons and hydrogen. All of the biomolecules are organic. Organic molecules may also contain nitrogen, oxygen, phosphorus, and sulfur. Inorganic nutrients may contain carbon and hydrogen, but generally do not contain carbon-hydrogen bonds like you see in organic molecules. Examples of inorganic molecules are salt, carbon dioxide, and water. They may contain either carbon or hydrogen, but not carbon bound to hydrogen. There are many factors that influence microbial growth, such as nutrients, temperature, pH, etc. Now we will discuss a few of these. Microorganisms can survive within a specific temperature range, and within that range are 3 cardinal temperatures. The minimum temperature is the lowest temperature at which an organism can survive, and the maximum temperature is the highest temperature at which it can survive. Microorganisms cannot survive at temperatures above or below the minimum and maximum temperatures. The optimum temperature is that which the organisms have the highest growth rate. For example, human pathogens can grow at room temperature, which is about 25 degrees Celsius, but they grow best at 37 degrees Celsius, which is human body temperature. Some organisms have a narrow temperature range and others a broad range. Based on the cardinal temperature range, organisms can be classified as psychrophiles, psychotrophs, mesophiles, thermophiles, and extreme thermophiles. You should know the temperature ranges for each group and be able to explain or identify each. Do not confuse psychotrophs with psychrophiles. Psychotrophs grow best at temperatures above 20 degrees Celsius, and because of this, they can cause problems with food spoilage. Psychrophiles are cold-loving organisms and grow best at temperatures below 20 degrees Celsius. They are found in areas that are permanently cold, like in the polar regions and in the depths of the sea. Mesophiles grow at moderate temperatures. Human pathogens are part of this group because the optimum growth temperature for mesophiles is about 37 degrees, human body temperature. Thermophiles grow best at temperatures above 45 degrees and have an optimum temperature of about 70 degrees Celsius. These types of organisms are found in environments like volcanoes and hot springs. Extreme thermophiles can grow at even higher temperatures, where their optimum growth is far above 80 degrees Celsius. This group only includes those from the Archaea domain. Organisms that use oxygen must have a way to protect themselves from the toxic byproducts that form during aerobic cellular respiration. These toxic forms of oxygen are singlet oxygen, superoxide ion, and also hydroxyl radicals. Organisms that can use oxygen must also produce enzymes that can break down the toxic byproducts. Enzymes like superoxide dismutase, catalase, and glutathione peroxidase are three enzymes that convert toxic oxygen into non-toxic molecules. Organisms that do not make these enzymes cannot use oxygen because the toxic byproducts would kill them. These types of organisms are anaerobes. This figure shows the toxic oxygen species that are formed during aerobic respiration with oxygen. The first toxic molecule that is formed from use of oxygen is the superoxide anion, which can cause major cell damage. Organisms that produce the superoxide dismutase enzyme can convert the superoxide anion to hydrogen peroxide. Some organisms also produce the enzymes catalase and glutathione peroxidase that can convert the hydrogen peroxide to water and oxygen, two perfectly non-toxic molecules. Both the superoxide ion and the hydrogen peroxide can cause damage to lipids, proteins, carbohydrates, and nucleic acid, all of the biomolecules that make up the cell. Organisms can also be grouped into categories based on the effect of oxygen on their growth, which is directly related to whether or not they produce one or all of the enzymes mentioned in the previous slide. You should become very familiar with this slide and be able to identify the group based on the effect of oxygen on growth, the level at which it will grow within a tube of solid medium, and the explanation for the growth patterns based on the enzymes that the organisms are able to produce. Anaerobes are microbes that must grow without oxygen, so they must be incubated in containers or equipment protected from oxygen. These organisms can be grown in a growth chamber, which is pictured on the left. Mixtures of gases like nitrogen, hydrogen, and carbon dioxide are pumped into the chamber, and oxygen is pulled out to keep the environment inside anaerobic. Organisms that can tolerate oxygen at low levels can be grown in an anaerobic jar, pictured on the right. A chemical packet is placed inside of the jar, and when the jar is closed, the environment inside becomes anaerobic. Organisms can also be grouped based on their pH requirement. The pH scale ranges from 0 to 14. Below 7, pH is acidic, and above 7, the pH is alkaline. pH 7 is considered neutral. This figure shows some common items that we should all be familiar with. Organisms that require an acidic environment are considered acidophiles. Alkalinophiles require basic or alkaline pH, and neutrophiles prefer a near neutral pH somewhere between 6 and 8. Tonicity is the ability of the extracellular fluid to make water move in or out of the cell, and it is directly related to osmolarity, the concentration of solutes in a solution. When we discuss the movement of water, we always refer to the concentration of the extracellular fluid compared to the inside of the cell. Water generally moves in the direction where the osmolarity is the highest. In the case of a hypertonic solution, the external environment has higher osmolarity than inside the cell. So water will move out of the cell, and if enough water moves out, the cells will shrink. The opposite occurs when the cells are placed in a hypotonic solution. The osmolarity is higher inside than outside in the external environment, and water moves into the cells. This causes them to swell and possibly burst. In an isotonic environment, the concentration of solutes is the same inside and outside of the cell. There is no net movement of water towards any one direction. Most microbes exist under hypotonic or isotonic conditions, but there are organisms like halophiles that require high concentration of salt, and others that don't require high concentrations of salt, but can tolerate those conditions. Pictured here is an aerial view of salt evaporation ponds. These man-made ponds are filled with organisms, and the types of organisms depend on their tolerance to the level of salt in the pond. The different colors are due to pigments produced by the halophiles. We've discussed different environmental factors that influence growth. Now let's take a look at how organisms grow at the individual cell level and at the population level. Bacterial cells divide by binary fission. The original cell doubles in size when it produces all of the components required for the new cell, including the chromosome. After the new chromosome is made, each of them migrate to opposite sides of the enlarged cell, and the cell membrane starts to separate the contents until the membrane is formed around each of the cells, and they eventually split into two. At that point, the cell cycle is complete. The time required for a complete cell cycle is called the generation or doubling time. It is different for different species of organisms, and the rate at which the cell cycle takes place is greatly influenced by environmental factors like temperature, pH, and osmolarity. Also, the availability of nutrients, to name a few. Generation times can vary from minutes to days. For example, E. coli cells complete a cell cycle once every 20 minutes or so when grown in rich medium at 37 degrees Celsius. But when grown at 25 degrees Celsius or in conditions where nutrients are low, the growth rate can slow to 12 to 24 hours. At the end of each cell cycle, the population increases by a factor of 2 because each cell makes one more cell. This is called exponential growth. This chart shows exponential growth when starting with a single cell. After the first generation, 2 cells result. Each of those cells divide by binary fission, and 2 more cells are produced, and so on and so forth. There is an equation we can use to calculate population growth, where N of F is the total number of cells in the population, N of I is the starting number of cells, N lowercase n represents the number of generations, and 2 to the nth power is the number of cells in that generation. If we start with 5 cells, Calculate how many cells there will be after 2 hours if the generation or doubling time is 30 minutes. So if we look at the number of generations, we can see that if the cells divide every 30 minutes, that will give us 4 generations in 2 hours. So 2 to the n is 2 to the 4. N of I is 5 because we're starting with 5 cells. And then if we plug the numbers into the equation, we have N of F, or the total number of cells in the population, equals 5 times 2 to the 4th power. This equals 80 cells. Pictured here is a population growth curve. If we monitor a population of cells growing in a tube of broth, we see that there are 4 stages. These 4 distinct stages can last longer or shorter amounts of time based on the environmental conditions and the species. The green in the tubes represents live cells, and the brown represents cells that have died and have settled to the bottom of the tubes. The first stage is the lag phase. This is a period of adjustment. The cells are adjusting to the new environmental conditions of the growth medium, and they are becoming larger in size, preparing new cellular components before they begin to divide. At this stage, there is very little cell division. In the exponential phase, cells are dividing at their maximum rate and will continue to grow at a high rate as long as there are adequate nutrients and a favorable environment for growth. This stage is also termed the logarithmic growth phase. When the growth medium starts to accumulate metabolic waste products and the nutrients and oxygen start to decrease, some cells start to die at the same rate other cells are dividing. This is represented in the bottom of the tube where you start to see the brown color. This is the stationary phase, where growth of the population has reached a plateau. The final phase is the death phase. It is also an exponential phase because more cells are dying than dividing, and this is due to the further buildup of waste products and the lack of nutrients. You can also see that there is even more brown at the bottom of the tube. We can measure population growth in several different ways. The most common method is by using a spectrophotometer. This instrument measures absorbance of light. On the left are 2 tubes. One tube is plain nutrient broth, and the other tube has a sample of a culture. The tube of broth is used for comparison. On the right, we see that when the broth tube is placed into the spectrophotometer, almost all of the light shines through, and very little is absorbed by the broth. But when the culture is measured, almost all of the light is absorbed by the cells. And you can see the difference in absorbance in looking at the numbers. The advantage to using a spectrophotometer to measure growth is that it's very quick and easy. All it takes is for you to put the tube into the spectrophotometer and wait for the reading. But this method does not distinguish live cells from dead cells. We can also measure growth using a viable colony count. Cultures are grown in tubes, and samples are taken out of the tubes at different time points and spread onto agar plates. The plates are then incubated and the colonies are counted. Only cells that are alive will develop into colonies. But this method is very time-consuming and expensive because you must use lots of broth in the tube for the tubes and also lots of agar plates when spreading the samples. Why is it even important to measure population growth? There are many reasons. When researching new antimicrobials, scientists want to see how effective the drug is at preventing growth during that exponential phase, when cells grow the fastest. And the goal is to accelerate the death phase. In the graph shown here, we see two different strains of E. coli that were grown in medium containing an antibiotic. The normal strain of E. coli did not grow, but the strain that is resistant to the antibiotic grew with no problem. We also study population growth of pathogenic organisms. We can mimic conditions that the organism may face in the human body and monitor growth to see how fast it might grow during an infection. We also monitor growth in food microbiology to keep our food safe from pathogens that may harm us and from microbes that may cause diseases in agricultural crops. Use this summary slide as a guide while studying this lecture.